Hello everyone. My name's Chloe Furst and it's my honour to chair today's Adelaide Biomed City Minireview research session on CALHN neurodegenerative disorders. Before we begin, I'd like to formally acknowledge that we are meeting today on the traditional lands of the Kaurna people and they remain the traditional custodians of the land, and we pay respect to the elders past, present and emerging. So following our speakers' presentations, there will be time for questions. So if the audience could please type any questions you may have into the question and answer section of the Zoom window, and these will be asked of the speakers at the end. I'd also just like to advise everyone that the session is being recorded and will be available on the ABMC website for watching at a later date. I'd now like to welcome Dr. Cathy Short. Dr. Short graduated from the University of Adelaide Medical School in 1995 and went on to receive her FRACP in Neurology and General Medicine in 2003. She's trained at both the Royal Adelaide and Queen Elizabeth hospitals. She's developed a special interest in behavioural neurology and the dementias. She's been working as a neurology staff specialist in the Queen Elizabeth Hospital Memory Disorders Unit since 2003 and has also been actively involved in more than 60 clinical drug trials for Alzheimer's disease and other dementias. Thank you, Dr. Short.
Thanks, Chloe. So I've got a very brief amount of time to tell you about what we do in CALHN memory trials. I just wanted to start quickly by running through some quick dementia statistics. Someone in the world develops dementia every 3 seconds. It's the second leading cause of death in Australians and leading cause of death in Australian women. In 2020, there were over 55 million people worldwide living with dementia, and this doubles every 20 years. We're expecting 139 million in 2050. And the annual global cost of dementia is $1.3 trillion US, and again expected to rise to 2.8 trillion by 2050. So dementia is an umbrella term. A lot of people use the term dementia and Alzheimer's interchangeably, which is not technically correct, but there are lots of different types of dementia that sit under this umbrella. Alzheimer's is the commonest form of dementia, and this is where the main focus is in the memory service and memory trials. This is just a little schematic from a paper from Jill Livingston in 2020, which looks at risk factors for dementia. And we can see from this schematic that about 40% of risk factors for dementia are potentially modifiable, but there's 60% where the risk is unknown. It's a complex multifactorial etiology for Alzheimer's disease. It's a complex interaction between genetic, environmental and lifestyle factors. Two major proteins involved are tau and beta amyloid. They're the pathological hallmarks. Amyloid beta accumulates early and is thought to drive the pathology of the disease. And this is the main reason there's been a big focus on the amyloid cascade hypothesis, and neurofibrillary tangles are very closely associated with cognitive decline and only emerge once the amyloid is present. Neuroinflammation triggered by innate immunity, as well as oxidative stress and mitochondrial dysfunction are also thought to contribute. In terms of therapeutic targets, this is again a little schematic about where the main focus for targets in Alzheimer's are. Majority of the work is being done around amyloid-based strategies, and this is sort of where we're focusing mostly in our clinical trials. There's also work being done on tau-based therapies, and these are more symptomatic type treatments, and the cholinesterase inhibitors are our current standard treatments for Alzheimer's disease. This is another schematic which I think gives a good idea of where things are in terms of the drug development pipeline for Alzheimer's disease. This was published by Jeff Cummings last year. And the main areas of research are differentiated into disease modifying and symptomatic, and then disease modifying biologics and small molecules. And we go phase 1, phase 2 and phase 3. These are all the different mechanisms of action. And this is the space that we're mostly focusing on in our memory trials unit. So phase 3, disease-modifying anti-amyloids. And in fact, we're doing trials in both gantenerumab and lecanemab. So CALHN memory trials. So memory trials at the Queen Elizabeth first started under Neurology in 1996, and it was set up by Dr. Karen Boundy, a neurologist who was involved in the first cholinesterase inhibitor trials in Australia. And Royal Adelaide memory trials were started under geriatrics in 2008 and was run by Dr. Jane Hecker and Rob Prowse, and the two units merged in 2017, and we've now currently got presence of trials across both sites, Royal Adelaide and Queen Liz. And as I mentioned, our main focus is on pharma-sponsored trials for Alzheimer's. Our current staff includes 2 neurologists, 2 geriatricians, trials manager. We've got 5 trial coordinators, 6 clinical trial raters who are mostly neuropsychology trained, and an admin officer. So currently we're recruiting for a phase 3 double-blind randomised controlled trial in lecanemab, excuse me, and this is in pre-symptomatic Alzheimer's disease. This is an anti-amyloid antibody. There's also going to be starting up soon the autonomy study at the Royal Adelaide, which is a phase 2 double-blind randomised controlled trial, which is an anti-tau monoclonal antibody. We're also currently recruiting for an FTD study, looking for people with progranulin mutations and giving them, again, potential access to this product called AL001, which is a monoclonal antibody to sortilin in FTD due to this mutation. We're also starting up at the Royal Adelaide, a phase 1 study looking at evaluating a cognitive platform to support the development of symptomatic therapies. We're also going to be starting another pre-symptomatic AD trial called Skyline, looking at gantenerumab, another anti-amyloid agent, XPro 1595, which is an selective anti-TNF agent. Looking at giving this in people with mild AD with evidence of inflammation on biomarkers. And there's going to be another Eli Lilly phase 2 study starting up soon, which is an oral OGA enzyme inhibitor. We've got ongoing trials with gantenerumab in prodromal and mild Alzheimer's disease. And we've also got ongoing trials with gantenerumab in mild to moderate Alzheimer's. We also have ongoing trials with lecanemab in prodromal and mild, and ongoing studies with Anavex, which is an oral selective sigma-1 receptor antagonist in mild Alzheimer's. I just quickly wanted to talk about ADNeT, which we are also involved in through memory trials in CALHN. So it's a coordinated approach to dementia research and clinical practice improvement. It's a multi-institutional consortium of dementia researchers and clinicians, and its funding is both philanthropic and NHMRC and NIDR. There's 3 pillars. CALHN Memory Trials is involved in all 3, registry, memory clinics, and also the ADNeT screens and trials. The one I wanted to talk to you today briefly about was the Screens and Trials Clinic, which is actually running at SAMRI. And the idea of this is to identify people who are at risk of developing Alzheimer's disease and channeling them into Alzheimer's disease trials for pre-symptomatic disease. We're also, through this clinic, looking at collecting data to contribute to ongoing observational research, looking at comparing amyloid and tau on PET scans in these people, and also assessing the degree of tau accumulation, comparing to amyloid burden, as well as atrophy, cognition, medical lifestyle factors to identify potentially modifiable factors that may delay dementia and subsequently inform clinical trial design. Very quickly, our screening criteria, these are pretty standard criteria for entry into any of our clinical trials. Age range roughly 50 to 85. You have to be able to speak English, more than 7 years of education to be able to be involved in the neuropsychology testing. And they also need a reliable caregiver. Exclusionary criteria are listed, obviously any significant mental health disorder, history of drug or alcohol abuse or dependence, cancer, or any significant or unstable medical condition, or if there's any contraindication to MRI. I think I might need to stop there because I think I'm running out of time. This is just our contact details at the Queen Elizabeth, Shelley, Casey, Zandy and Alexandra, and at the Royal Adelaide, Angela, Jane and Olivia. And I just wanted to thank the CALHN Memory Trials team.
Thanks very much, Dr. Short. So just briefly, why do you think ADNeT has been such an important step forward in dementia research?
So particularly for South Australia, ADNet's given us access to amyloid PET scans for people who want to assess what their risk is for developing Alzheimer's disease. As we know, amyloid can start to accumulate 15, even 20 years before people start to show symptoms of cognitive impairment. So it's been quite helpful from that point of view. And hopefully, in due course, we'll be able to produce our own amyloid tracer at SAMRI. There's been a few teething problems there. So currently we're still flying in tracer from Melbourne. But the aim is ultimately to be able to produce our own amyloid and tau tracers at SAMRI, so that ultimately we'll be able to do these tests on a private outpatient basis rather than just through research.
You briefly mentioned it, but can you elaborate more on the value of registry data for a condition like dementia?
So the registry data is very helpful in terms of identifying where there are areas of need around the country. So this registry is being done Australia-wide, and is collecting a wide range of demographic data on people with a new diagnosis of mild cognitive impairment and also Alzheimer's disease. So it really gives us a good snapshot of what's happening around the country and will hopefully help identify any gaps in funding and resources around the country.
And one final question, which is very broad. Other than, I guess, a cure for dementia, where do you hope dementia research is going to go in the next 5 years?
So I think in the next 5 years, it's very likely that we're going to see an anti-amyloid agent brought to market. There's already been approval for aducanumab in the States under somewhat controversial circumstances, but I think that's really going to open the door for a lot of other agents to be brought to market in the very near future. I see it as a disease that's probably going to be treated in several different ways. I don't think there's going to be one magic bullet. I think it's going to be a range of different treatments, probably cholinesterase inhibitors, anti-amyloid agents, possibly secretase inhibitors as well. So, yeah, I don't think it's going to be one single drug.
Brilliant. Thank you so much, Dr. Short.
That's all right. Thank you.
Okay, we might now move on to, it's my pleasure to introduce Dr. Thomas Kimber. So Dr. Kimber graduated from the University of Adelaide in 1989 and then undertook basic physician training at the Royal Adelaide Hospital. He's completed his neurology advanced training at the Royal Adelaide Hospital, Sir Gardner in Perth, and the Royal Free Hospital in London. He's completed a PhD, which concerned the effects of pallidotomy surgery on motor function in Parkinson's disease, which was awarded by the University of Adelaide in 2008. So in addition to his private practice, Dr. Kimber is a senior consultant neurologist at the Royal Adelaide Hospital and a clinical associate professor in the Department of Medicine at the University of Adelaide. He sees patients presenting with a wide range of neurological disorders, but his main clinical and academic areas of interest are Parkinson's disease and other movement disorders. So thank you, Dr. Kimber.
Thank you very much, Chloe. and thanks for the invitation to talk about the CALHN Neurology's program in Parkinson's disease research and research in related conditions. Apologies for the split screen. For those of you who are not overly familiar with Parkinson's disease, I'll give a bit of background, just as Cathy did, into this important condition. It is the second most common neurodegenerative condition after Alzheimer's disease, and nearly 100,000, I should say, Australians are affected. With this being a disease in which the risk increases with advancing age and with our aging population, but not only associated with that risk factor of aging, there is emerging a global pandemic of Parkinson's disease, and it's now estimated in the Western world that there's about a 1 in 15 lifetime risk of developing this condition. And while it does increase in incidence with advancing age, it's by no means reserved to older people, and an important minority of sufferers are diagnosed before the age of 50. The problems clinically that Parkinson's causes are seated in the underlying neuropathology of this condition. And I want to briefly talk about this as it impacts on where research is moving in this condition. The pathology of Parkinson's disease is that involving the synaptic protein alpha-synuclein. This protein has an important role normally in synaptic function and is usually present in a monomeric form. But in Parkinson's disease, it undergoes conformational change to toxic fibrils and oligomers, which then lead to neurodegeneration within a range of circuits within the central nervous system. And degeneration within both dopaminergic and non-dopaminergic circuits as a result of this pathology underlies, in general, the major motor and non-motor manifestations of the condition, respectively. The cause, or more correctly put, the causes of Parkinson's disease are still being established, but much now is understood about the underlying molecular basis for this condition. And suffice to say, there are a number of sites within neuronal cellular metabolism and function where abnormalities, be they at gene transcription level, protein expression level, within mitochondrial function, within lysosomal function, to name a few, can all converge in the common pathological endgame of Lewy body formation and neurodegeneration. So the CALHN Neurology Parkinson's Disease Research Unit was formed in 2002 under the leadership of then Head of Unit, Professor Philip Thompson. And its earliest activities, and this remains a significant focus of the unit now, was in involvement in multi-centre sponsored clinical trials, such as those listed here, primarily involving novel dopaminergic therapies, dopamine replacement therapies, to look at safety, tolerability, and efficacy in treating both motor and non-motor symptoms of Parkinson's disease. And these include studies of some of the device-assisted therapies for more severe disease. We've also been involved in an anti-tau monoclonal antibody study, unfortunately an unsuccessful one, for an currently untreatable related neurodegenerative disorder, progressive supranuclear palsy. We've also been a leader in several investigator-led observational studies, particularly focused on a clinical focus of our unit, which is device-assisted therapies for Parkinson's. And some of these are listed here. The first Australian longitudinal study of response and adherence to apomorphine infusion, and our initial experience with levodopa-carbidopa intestinal gel infusion for Parkinson's disease with our gastroenterology colleagues. You will notice that all of the studies I've mentioned primarily involve symptomatic therapies, trials of symptomatic therapies for symptoms of Parkinson's disease. And this reflects that, unfortunately, we're still in an era in Parkinson's where we have no proven disease-modifying or preventive therapies, and we're desperately in need of them. And this has informed a development in our research interests in the Parkinson's Research Unit, which is greater involvement in studies that are interested in potential disease-modifying therapies. And we're in our earlier stages of involvement in such studies, but we're proud to be involved in a current collaborative study called the Australian Parkinson's Mission Product, or the APM. The website is here for those of you who may be interested. And this is an Australian-led international collaboration of which we are one investigating centre. And this study is, the APM 001 is the inaugural study of this consortium, and it's looking at 3 repurposed medications, a gliptin, a beta agonist, and a calcium channel blocker, for all of which there are biochemical rationales to believe they may beneficially alter neuronal cellular processes that may potentially reduce the progression or slow the progression of Parkinson's disease. And these are being compared against placebo in this study, and results are eagerly awaited. In the time that remains, I want to talk to you about another research interest in which we are particularly excited at the moment, and that is a study we have running in fecal microbiota transplant, or FMT for short, as a potential therapy for Parkinson's disease. And we're doing this with our collaborators in the CALHN gastroenterology units and Biome Bank. And I'll tell you a bit more about that in a minute. A little bit of background, but some of you may be aware of the emerging interest of the gut microbiome in a range of different medical conditions, including neurodegenerative conditions. There is evidence that the gut microbiome may be integrally involved in the earliest pathology of Parkinson's disease, which there is some evidence to show may actually begin in the gut, and pathology may then travel in a retrograde fashion from the gut along the vagus nerve back to the central nervous system. And the gut microbiota may be involved in a number of different ways in the earliest pathological processes, perhaps by mediating intestinal wall inflammation and breach of the intestinal barrier to transfer of neuropathology across the gut membrane. There is also some evidence that the gut microbiota may be involved in the fluctuations in motor response to dopamine replacement therapy, particularly levodopa, that occur as patients with Parkinson's disease continue their disease course, where they oscillate between severe off motor function and on function, sometimes with involuntary movements called dyskinesias, in response to their medications. And there is evidence that a range of gut microbiota, including in fact H. pylori, interestingly, that we associate with peptic ulcer disease, may be involved in motor fluctuations. And finally, there is evidence from a recent randomized controlled trial for probiotics as being a potential treatment for one of the major non-motor symptoms and earliest non-motor symptoms of Parkinson's, that is constipation. So a lot of interest on the gut microbiome. So this study that I'll briefly mention is a collaboration between ourselves, CALHN gastroenterology, particularly Drs. Robert Bryant and Sam Costello, who are also integrally involved in Biome Bank. This is a South Australian entity that collects and produces in a biologically usable form donated fecal matter and microbiota for use in the treatment of, for example, gut diseases such as C. difficile colitis. We're also collaborating in this study with nuclear medicine colleagues, and it's supported by the Weeks Group. And this is a 6-month open label pilot study of fecal microbiota transplant, 12 Parkinson's patients. We've currently randomized, I believe, 7. So since this slide was produced, a few more. These are people experiencing fluctuations in their response to levodopa. And the principal goal being a pilot study is to establish safety and tolerability of FMT. But we have a number of primary and secondary endpoints looking at both motor and non-motor symptoms in Parkinson's disease that we hope will show us a signal to potential clinical benefit that we might hope to explore in a subsequent larger placebo-controlled study. So that's all I have time for. I would like to acknowledge my collaborators currently from the gastroenterology and nuclear medicine departments, and particularly the neurology colleagues with whom I work in the CALHN Neurology unit, particularly our Parkinson's clinical practice consultants, Heather Webb, Linda Huddy and Rebecca Krukam, my co-investigators as listed there, and my neurology colleagues more generally in the department for their referral of patients to our research program, and most particularly the patients and families who generously volunteer for our clinical trial program to try and find better treatments for this disorder. Thank you.
Thanks, Dr. Kimber. So just briefly, you've mentioned that obviously you've been doing a lot of work with your gastro colleagues. Do you think there are opportunities to partner with other craft groups or specialty groups, and in what particular areas do you think?
Yeah, I think absolutely. We are very interested and enjoy very much our collaborations with other professional groups in this condition. So Parkinson's is a condition which, as I mentioned, is way more than a neurological or at least a motor disorder, and involves a range of very disabling in many patients non-motor symptoms, urinary, gastrointestinal tract, sometimes pain and other sensory phenomena, autonomic dysfunction, to name a few. And so we are very interested in collaborating with other investigators who deal with Parkinson's disease. Geriatricians would be another group, Chloe, as well. If you told me I would have been collaborating in research in Parkinson's disease with gastroenterologists when I started working in Parkinson's disease, I would have, said, why on earth, that sounds crazy. But the way things have developed, we're dealing with a multi-system disorder with an ever-evolving concepts as to its pathogenesis and its progression. And the more we can collaborate, the better, I think.
Fantastic. Thank you very much. So we're sort of out of time today, so thank you all for attending what has been a hugely informative presentation. If the audience has any feedback or comments regarding the session format or presentations, they can be entered on the question and answer function for the next few minutes. Otherwise, please contact the ABMC via their website. As mentioned at the beginning, this session will be uploaded to the website in the coming days. And next Tuesday's presentation is on cancer in women, with a presentation by Prof. Martin Oler and Associate Professor Teresa Hickey, and chaired by Andrew Zanantino. So thank you very much for joining us today, and we'll see you next week. Have a good day. Bye.